So thank you for the invitation to speak in this series. And I'm going to present some of my work here at Cornell over the past few years. And I'm going to talk about the origin and genetics of Cran's anatomy and C4 anatomical specialization. So just some background. We as humans are not doing too well with our planet. You know, this is how we've been doing agriculture so far, is pretty much rip down nature and put in the plants that we think are pretty tasty. So this is not good for our ecosystem or the other animals on our planet. I just think this is a good one here, what we're doing to our planet. So humanity is going to face some really hard problems in the near future. We're going to have another billion people in the before 2025, and almost 9.5 billion by 2050. So we need more food. This increasing population is increasing demand. More meat consumption means more grain. Our agricultural lands that we do have in production are becoming degraded. We can't bring much more into production. There's nowhere to go with our lands. We have an increasing demand for bio-based energy, and that's going to come from plant biomass. We need to increase our biomass output. We have limited natural resources to support our current high-intensity agriculture, especially water, phosphorus, and fossil fuels, which also impacts nitrogen. So one of the radical solutions to increasing productivity per land basis is to actually modify photosynthetic capacity in plants. And the one we work on is actually using C4 to increase the photosynthetic output of plants. Well, why do we want to look at C4 and use C4? Well, when you see a field of plants growing next to each other with C3 and C4, it's pretty clear. In the foreground, we have rice, which is a C3, and this is 42 days after transplant. Right behind it is Echinocloa, which is a C4 grass, planted at the exact same time. Behind that is maize, another C4 plant, which is 44 days after germination. And the difference between rice and Echinocloa is almost a 50% increase in biomass. And that's attributed to the C4 process. And even in the back, the C4 maize, that's mostly due to domestication traits after C4. So the difference between Echinocloa and maize is mostly domestication with the C4 trait. So it's a pretty apparent why we want to do this, because these are all growing on the same land with the same inputs, but these are much more productive as far as biomass. So transferring the C4 into C3 crops is predicted that it could eventually boost yields by 50%. Now, just putting C4 into our crops won't get us instantaneous yields. It's going to take a lot of breeding afterwards at that point, as you've seen with the difference between Echinocloa and maize. Getting that into harvestable tissue is going to be another big challenge. So, but based on our current knowledge base, when I started this project, it was estimated by the C3, C4 working group that it would take up to 20 years and anywhere between $100 million and $1 billion to engineer C4 rice. So it's a large undertaking. And you know, when I present this at other meetings, especially in industry, they pretty much just laugh in our face. They said it's just impossible to do. So it's too complicated. But even with all its complications and time limitations and money, you know, the possible returns could overshadow the costs. Just not the 50% yield increase that would be on target for what we need for humanity's needs. You know, the 50% yield increase itself would account for about $60+ billion in crop value per year. That's about an estimate on our current values. But also a savings of almost $7 billion a year reduced input costs, which is the water use efficiency. C4 is more water use efficient, and nitrogen use efficient. The big problem so far is engineering the specific architecture and the cell-cell interactions that are needed for C4. So it was not understood at this point, because despite 30 years of research, there are no known genes identified that function in Cran's anatomy and setting up the C4 anatomical traits. So this is where I started on the project, and this is what was given to me as like a large task ahead. So I wanted to know, well, how did nature do it to begin with? You know, how did it evolve? Maybe that'll give me a blueprint for how you could engineer it. So I wanted to start with the evolution of C4 in plants. It turns out that Kranz type C4 evolved about 66 times independently. Now this is like one of the most remarkable examples of convergent evolution in life itself, 66 times the exact same way, or similar ways. So it has to be easy. If it is 66 times that we know of evolved, it has to be just a few changes that are needed to get there. And every time it evolves, it just pops up fully formed. So there's little evidence that there was a slow evolutionary progression towards Kranz anatomy each time it evolved. It just pops up fully formed. But how can we reconcile this with Darwinian evolution, which is more like Goldschmidt's hopeful monsters, where spontaneous complexity just arises? So it was really another huge mystery is how such a complex system just pops up. So when we look across the plant kingdom, we see that C4 occurs both in monocots and dicots. So there's a few of the dicot species, including Cleome, Flavaria, and their Caryophyllales, which have a lot of the C4s popping up again and again. But it looks more like a random distribution through the rest of the dicots. But if we look, I want to come back to this later in my talk, just take this into account, that there appears to be some ancient preconditioning event in the grasses, where there's a large proportion of the grasses that are C4. So it occurs all throughout plants, and there's something maybe similar between all these plants that's giving rise to C4. So what's so special about C4? We'll get into little nuts and the bolts, and I promise this will be the only slide in biochemistry on C4 I really present this time. So most C3 plants only have one cell type that fixes carbon dioxide, and that's the mesophyll cells. And in the mesophyll cells, carbon dioxide diffuses into the mesophyll cell and is fixed directly by rubisco to make a C3 product, 3-phosphoglycerate, which then goes into the Calvin cycle. Now this only takes about 18 ATP to do. But in C4, CO2 is fixed twice. Once in a mesophyll cell, and then it's shuttled into a bundle sheath cell and fixed rubisco there. And in C4, rubisco's sequestered in the bundle sheath cell away from the initial fixation. So CO2 comes into the mesophyll cell, is turned into carbonic acid, and immediately into oxaloacetate, and into the first stable product, which is malate, which also can be aspartate. Now, malate and aspartate diffuse through plasmodesmata from the mesophyll cell to the bundle sheath cell, where the CO2 is released from the malate to produce CO2 and pyruvate. Pyruvate goes back into the mesophyll cell to start the cycle over. But what this cycle does is take the CO2 and concentrate it around rubisco in the bundle sheath cell. Now this takes about 30 ATP. It's actually more costly. Well, you say, well, why would you want to do something more costly in general? Nature doesn't like to do that. But the problem is rubisco can also use oxygen as a substrate. And when it uses oxygen, it makes this 2-phosphoglycolate, which is toxic to the cell and takes a lot of energy to reclaim. So sequestering rubisco into this cell type and concentrating the CO2 reduces photorespiration, excludes CO2 from rubisco, saturates rubisco with CO2 to achieve maximum catalytic efficiency. And the C4 plants are more water and nitrogen efficient because they're more efficient at using CO2. And they're more nitrogen efficient because a lot of nitrogen in the cell is tied up in rubisco, because it takes a lot of rubisco to perform photosynthesis. So if you don't have to use as much rubisco, your nitrogen use efficiency goes up. And overall, it increases radiation use efficiency per unit area. And this is what we're looking after as far as engineering C4. And the most striking thing I found out about C4 is although C4 only accounts for about 3% of the total vascular plants, it's responsible for about 25% of the total terrestrial photosynthesis on Earth. So it is not a small portion. So it's accounting for a lot of what's going on. So the basic mechanism of C4 requires a specialized architecture called Kranz anatomy. So some basics of the cell type and what's going on. So Kranz is German for wreath, which is the structure around the vein. And it is a single cell layer, the bundle sheath, that surrounds a vascular core. The bundle sheath cells are usually suberized, which is like a waxy coating that limits gas and solute exchange. Bundle sheath cells preferentially accumulate starch. Mesophyll can accumulate starch when the phloem is blocked and carbohydrates backed up, but it seems to have a preference for the bundle sheath. And there's an increased vein density in the C4s, which leads to a 1-to-1 ratio of bundle sheath to mesophyll, where you get this vascular core, bundle sheath cell, mesophyll, mesophyll, bundle sheath cell, and then another vascular core. Now, there's also a difference in the chloroplast between the bundle sheath and the mesophyll. So in the bundle sheath, have a specialized chloroplast type, which only has photosystem 1. It lacks photosystem 2, so it doesn't have grana. But the mesophyll plastids look almost normal. They have the stacked grana. They have both photosystem 1 and 2. And there's still unknown how this difference between the plastids arise in C3 and C4. And again, the bundle sheath preferentially accumulates starch. And also, between these two cell types, there's abundant and specialized plasmidesmata that allow for the transport of the metabolic shuffling between the two cells. And if you can't really see here, but I'll come back to it later, the mesophyll side has these specific sphincters just on the mesophyll side. And that'll come into play later when we look at cell identity. So we want to engineer C4 similar to maize into C3 rice. So how does rice compare to the C4 maize? Well, again, in maize we have high vein density, and you have the 1-to-1 ratio of the bundle sheath, mesophyll, mesophyll, bundle sheath, and the bundle sheaths are photosynthetic. But in rice, vein density is reduced. You almost have 7 to 8 mesophyll cells between vascular cores. And rice does have a bundle sheath-like structure and also a mesotome sheath, which are not continuous around the vascular core. But both the mesotome sheath and the vascular bundle sheath are not photosynthetic. And they have undifferentiated cells, plastids in those cells. So there's a similar architecture here, but not exactly what we need for C4. So we'll come back to this in the end when I look at the evolution of the Kranz anatomy in grasses. But there's similarities here, but there's still a lot of differences. And when we look at engineering C4s, how do we increase the vein density? How do we get the proper cell-cell interactions needed between the bundle sheath and mesophyll into rice? And also activate the photosynthetic system in the bundle sheath. So there's still a lot we didn't know. So this vein spacing in dicots between C3 and C4 seems to be worked out pretty well. The difference between the C3 Flavaria species and a closely related C4 Flavaria species shows that the vein density itself only changes with the minor veins. It's not the major veins that are changing, it's just the smallest class of veins. So here we have like 3rd order, 4th order, 5th order, and those are the larger vein classes. But in the C4, it's the addition of an extra vein class, the 7th order here, and more of them that are leading to the increased vein density and the 1-to-1 cell ratio we saw in C4. And if you look over developmental time, the major vein classes, 1, 2, 3 through 4, all pretty much develop the same rate, same time. But it's only those minor vein classes that have changed, and they appear to rapidly develop once these major vein classes have formed. So it's just something special about the minor vein class in the C4 that's differing. So it was unknown in monocots what is leading to this increased vein density and cell-cell interactions. So that's where I wanted to start the project. So to start, I figured it's a developmental problem. Let's look at development. How do the veins develop? How did the bundle sheath develop? And one of the earliest markers for vascular development in any plant is the pin-auxin efflux carrier, which demarcates the cells that will become the vasculature. And you can see, you can observe the entire progression of the vascular core through the bundle sheath with that marker. So what I did is took pin YFP plants, maize, and if you take a cross section right through the base of the plant, you will actually get the developing leaves So here we have the developing leaves encircling each other. And in the younger leaves, we see that the veins actually start out as a procambial strand, a meristematic strand through the leaf. And then the major veins form at the base and become punctate from this procambial strand into intermediate and major veins that are evenly spaced. So the auxin gradients are leading to the demarcation of major veins at the base. But what about the minor veins? So the major veins form at the base. What's happening with the minor veins? Well, once the major veins form and have almost completed their developmental progression, they just need to enlarge, the minor veins start from the tip of the leaf and intercalate the major veins down towards the base. So major veins form at the base, and minor veins form at the tip and start intercalating the major veins. And it appears that the major veins provide the positional cues for the minor veins. And also, minor veins are predicted to form an auxin minima. So the expression of PIN in these veins is sucking out the auxin, providing the signals and the initiation of the minor veins. So then I figured, well, okay, we know how minor veins form, but this seems like a pretty simple model for vein formation in maize because it's almost one cell giving rise to a whole vascular unit with the bundle sheath. So I wanted to take a look at the minor vein itself. And if we look at PIN expression, this is a developing minor vein, and this is the tip. This is going towards the base, and this is towards the tip. And this is a stylized model of what's happening here with the PIN marker. We see that bundle sheath is actually a pretty early event, as early as the vein itself, where the PIN demarcates a single cell that's going to become the vascular unit, and the cell enlarges, divides, but also bundle sheath cells are recruited. It's not just from division, it's both a process of division and recruitment that give rise to the early bundle sheath. Once the bundle sheath are established, they divide and form clonal patches along the vascular unit as the internal vascular core differentiates. So, okay, we see the minor veins are formed later than the major veins, and the bundle sheath come off really early. And this is all before the transverse veins connect the parallel veins together. So the next major thing is that when I started this, it was unknown that it was actually not reported in any C3 plants that there's PIN expression in bundle sheath in the leaf blades. So it seems to be something specific about C4 plants that they are able to express PIN in a polarly localized fashion in a high expression pattern in the bundle sheath. Something specific about C4 and auxin. And we'll come back to this in a bit, but this was a new finding that C4 plants are able to conduct auxin and PIN. So summary of PIN and vein development. Lateral and indeterminate veins are derived from procambial strand. Minor veins form the tip, go to the base, and they're guided by the auxin gradients of the large lateral and intermediate veins. And PIN shows high expression and polar localization in bundle sheath cells. So this is when I came back to start thinking about evolution. Well, I've seen development, but is it likely that these cells develop from a unique process every time it arises, or is it something similar somewhere else in the plant that's going on that maybe C4 just hijacked another developmental process into its development? Maybe there's another tissue type that's similar, that it doesn't have to de novo produce this cell type, but can just borrow it from somewhere else. So I started looking through plant anatomy and seeing if there's something similar out other than a plant. And I went to ESA, 1956, Plant Anatomy, second edition, and found two statements that just kind of changed everything for the project. So here she says the bundle sheath in angiosperms is an endodermis. She says that a lot of the bundle sheath have properties of the endodermis like in the stems and roots. And the bundle sheath may also be a starch sheath. Now, a starch sheath is the endodermis in the stems and petioles. And the starch in the starch sheath gives gravity cues for the plant to maintain its upright position. And this was before C4 was known. She just described all of the starch sheaths, whether it be in the leaf or in the stems as an endodermis. And it turns out the ones she said that have starch sheath in the leaf were C4 plants, such as maize and sorghum. So this led to a new hypothesis. Well, Kranz anatomy, or Kranz type C4, may be derived from a conserved tissue in all angiosperms. And Kranz anatomy and C4 are derived from the projection of the endodermal identity onto the bundle sheath within the photosynthetic tissue. And it results in a synergistic interaction between the photosynthetic tissue and the endodermis. This would explain why, again and again, it arises fully formed, because instead of a slow progression of gene by gene, it's two programs interacting that are already inherent in the plant. We know that starch preferentially accumulates in the bundle sheath because it may be adapted from a starch sheath in the stem and petiole. And is it possible that the bundle sheath plastids are photosynthetic amyloplasts? They have a mixed identity. And this is why the dimorphic chloroplasts arise each time. Stubrin is also a characteristic of the endodermis, and we find it always associated with the bundle sheath. When you read the C4 literature, people are like, you need that apoplastic barrier, but it may just be a pleiotropic effect of the endodermis and may not actually be needed. And in root stems and petioles, the endodermis is an auxin-conducting tissue with high pin expression. And this may also explain the shift in vein density always seen in C4. It's not because it's required. It's because if you have an endodermis pulling more auxin out of the leaves, you initiate more auxin minima, initiate more minor veins. So the next big thing with this hypothesis is that the endodermis is actually primed with C4 metabolism. This is a paper from 2002 in Nature from Hibbert and Quick, showing that all the metabolism needed for C4 is present in the stems and petioles. Well, what's in a stem and petiole but the endodermis, parenchyma cells, and an outer layer of photosynthetic cells? So if it's already inherent in this tissue, maybe that's what's giving rise to this C4 metabolic shuffling. So the general hypothesis is that I'm using a dicot model here, that the endodermis stops right in the petiole before it hits the leaf blade in a C3 plant. And in C3, you have the vascular core extend from the petiole all the way out into the leaf, and it's coated with the C3 bundle sheath, which is just the same as the C3 mesophyll cell. But with the C4, all that's happening is that endodermal program, which is normally repressed, just escapes repression and gets projected out into the leaf blade. It sets up Kranz anatomy and preconditions the metabolism for C4. It's a simple change that leads to all that complexity that we've seen associated with C4. And this may be why that it can be reconciled with Darwinian evolution again, because it is a small change. And it can happen in any of the plant species. So the synergistic interaction may come from the mesophyll cell photosynthetic program projecting into the endodermis, activating photosynthesis, which is not normally photosynthetic. So here's the vascular core, bundle sheath, mesophyll cell. So the photosynthetic program invades the endodermis, and the endodermis projects a cortex-like identity onto the photosynthetic cell next to it, which gives rise to the CO2 metabolic shuffling. Now the endodermis and the cortex and other parts of plants are respiring tissues. They create CO2 and then have to get rid of it. They don't have a ready access for it. So this may just be the waste management system somewhere else in a plant being hijacked into the photosynthetic system. So if this is correct, just projecting the endodermal program into C3 leaves may be one way to engineer C4, or at least precondition it. Now, when I started the project, it was estimated that it would take 250 transgenic constructs, a mini chromosome, and 20 years to engineer. But if we can follow evolution and get it to do most of the work for us, I'd rather take this route. So where do we go from here? That's a good evolutionary argument, but we can't really go back in time and watch it happen. So I figured if this is a conserved tissue type, it should be regulated by the same genes as well. So what genes are we looking for? Well, when I looked at the minor veins again, I said, well, that really looks like a root. It just looks like roots developing in the leaf. So I wanted to go after the genes that regulate the endodermal program in the roots. So here's a stylized model of the Arabidopsis root tip. So we have the stele, which is the internal vasculature, the endodermis, which is in purple here, and the cortex in blue. So the genes for this are known, but they have a complex interaction. So here's the cell types for the stele, endodermis, and cortex. Now it's known that there's a gene that's expressed in the stele, and the protein moves from the stele, which is called short root, into the endodermis, and activates another gene called scarecrow. Now scarecrow actually stops short root movement, defines one cell layer, interacts with short root to activate other genes that are called jackdaw and magpie, to give rise to endodermis identity, and also it's still unclear, but it's likely that this complex moves into the cortex and activates more genes like the jackdaw and indeterminate-like. Now, in Arabidopsis, you call it jackdaw, magpie, and nutcracker, but they're all members of the indeterminate growth family that was originally cloned from maize. And these function with scarecrow and short root to establish these cell types. So this is where I started. And the first gene I wanted to work on was the scarecrow, because it defines the endodermis and defines that one cell layer. So in Arabidopsis, the scarecrow mutant has altered starch sheaths in the petiole, and when you have a mutant cell layer, it does not accumulate starch. So starch is a good indicator of endodermal identity. So throughout the talk, this is what I'll be using as far as endodermal identity is starch-stained leaves. And in maize, the ortholog of scarecrow has already been shown to be expressed in the early stages of vascular development. And it looks like it's right after pin turns on. So here's an early leaf. And at the base is when you have that procambial strand that then becomes punctate. And it seems to be turned on the major veins right after it becomes punctate and off those. Again, scarecrow in maize, this has an expression pattern that fits. This is a heat map generated by Tom Bertinel's lab of the developmental gradient in maize. So at the base of the leaf, it's still pretty much undifferentiated, and it goes towards a fully mature differentiated tip. And it looks like scarecrow is highly expressed at the base and then turns off as the leaf matures, and also has a mesophyll-enriched expression pattern. So with that, I looked for mutant alleles, and I was able to isolate 2 transposon insertions into this gene. And both occur in the first exon, so it looks like we're fully disrupting gene function with these 2 alleles. So I began characterizing those genes, or those mutant alleles. And the first thing we want to do is, well, is it conserved in the root function, as it is in the Arabidopsis? So in wild-type maize roots, we have the endodermal layer here, with its characteristic Casparian strip, which is the accumulation of suberin. So, but in the mutants, that Casparian strip is lost. Now in Arabidopsis, you actually lose the cortex cell as well, but we didn't find that here in the scarecrow mutants. And Arabidopsis and maize roots are pretty different as well. We didn't expect full conservation, but it does look like it's impacting endodermal identity in roots. But what about the leaves? So in wild-type plants, we have these nice vascular cores surrounded by one single layer bundle sheath, and red is chlorophyll fluorescence. And right outside the bundle sheath, we have a dense mesophyll cells. But in the mutants, we have very disrupted Kranz anatomy and a spongy-like mesophyll identity. So this is our first indication that we're impacting Kranz anatomy by disrupting this gene. So we have extracellular layers. We have some genes, or some cells, that don't have the densely packed chloroplasts like normal bundle sheaths. So when we start looking for specific mutations in Kranz anatomy, we find many. The scarecrow mutants produce extra bundle sheath cell files in many of the veins. So here we have one vein vascular unit in the middle, and on one side it's producing an extra cell file here, and an extra 4 on the other side. Now this is consistent with the hypothesized role of it stopping the short root signal from propagating out and recruiting more cells into the endodermal program. And they usually occur in the transverse vein. So these top 2 cells are usually recruited into come into the bundle sheath identity from division. But the ones in the transverse vein, the lateral sides, are usually from recruitment when I looked at development. So again, this is a major feature of Cran's anatomy that's distorted with this gene. The mutants have defective minor veins. So in wild type, we have a nice regular parallel pattern. But in the mutants, the minor veins have a really hard time forming. They usually crash into the veins next to it. They're having trouble forming in those auxin gradients, which may be distorted from distorting the endodermal identity. A lot of times, the minor veins terminate abruptly or get absorbed into the transverse veins that usually connect the two parallel veins together. And in the more severe cases, you have a loss of the minor veins altogether. So it's really impacting those minor veins, which give rise to the increased vein density. So, and also with the minor and major veins, the veins frequently collide. So the vascular cores don't collide, but you get these collision of bundle sheaths where they merge into a continuous structure that displaces mesophyll. So you get a vascular core, bundle sheath, bundle sheath, bundle sheath, vascular core. And it appears that bundle sheath formation is before the mesophyll formation. So if you disrupt that, you're disrupting the whole Cran's anatomy process. The next interesting thing is scarecrow also regulates the identity of the bundle sheath itself. So starch accumulation, we saw from the Arabidopsis mutant, is a good indicator of endodermal identity. So we used that in maize and found, especially in the M1 allele, which seems more severe, that we lose identity of some of these bundle sheath cells where they failed to accumulate starch. Now here's a major vein where the top and bottom cells have not accumulated starch. And there's some veins that don't accumulate starch at all. And this is a blown up picture of that showing the transverse veins connecting to it, but there's no starch. But the interesting thing is, those bundle sheath-like structures are still there. Even though it has not gained identity by accumulating starch, there's still the cell structure there. And it's striking how much it kind of resembles the mesotome vascular bundle sheath in rice. So we'll come back to that again, which you can lose identity but still get that structure. So when we looked at those cells that lost the identity, we're like, well, did it revert to a C3 chloroplast? Did it revert to a mesophyll-like identity? So we isolated some of those cells that lost identity and did some TEM analysis to look at the plastid structure and found out they remain undifferentiated. They don't take on a C3 or mesophyll-like identity, but scarecrow itself is required to set up that plastid identity. So, and also, we found that, again, the plasmodesmata, which are required for the cell-cell interactions between the two, are also altered. So in wild type, you have the bundle sheath and mesophyll, plasmodesmata that connect them, and only sphincters on the mesophyll side. But in the mutants, we're finding that the sphincters are actually on both sides for the undifferentiated plastid mesophyll, suggesting that there's you've disrupted the identity of the bundle sheath itself. So overall, the combination of these effects reduces vein density in the plant. And there's, these mutants are a little bit temperature sensitive, so when we grow them in the more severe temperature, we get down to a vein density similar to rice, actually. So there's a great loss in vein density, and especially we think it's just the loss of the minor veins of themselves. So the overall effects on Cran's anatomy are significant. So when we looked at the vein formation, the lateral vein number, vein density, bundle sheath cells, and starches cells are greatly distorted in both mutants, especially in the M1. We think what's happening is the M1 is creating a poison protein that's taken down the whole complex, and it's more severe than M2. Now these occur in wild type, but we think it's because it's a stochastic signaling process. You do get starchless cells, sometimes you get extra bundle sheath cells. It's not perfect every time, but we're greatly distorting it with the mutants. But even though we're really distorting Cran's anatomy, we didn't really distort C3 or C4 metabolism. So there's no change in isotope discrimination between the two, which is a signature of C4. So it doesn't revert to a C3-like state. So we've disrupted anatomy, but not really taken out the physiological state yet. So, well, we've shown you scarecrow regulating the endodermis, but what about the other genes? What are they doing? So I wanted to know what's the role of short root and the endodermal-like transcription factors in anatomy. So if this is a true conserved pathway, the rest of the genes should be conserved. So I was able to isolate not only the 2 in scarecrow, but 1 in the ortholog short root, 5 alleles in scarecrow-like 3, and the paralog of short root, which is short root on chromosome 7, 1 there. Now, indeterminate 1 has already been described in maize as a flowering time mutant, and there's multiple alleles available for that. And it's able also to isolate 5 other indeterminate-like genes and 1 to 2 alleles each. I didn't have time today, but I started the forward genetics approach where I identified a Kranz mutant called LSN. It turned out to be a non-coding RNA. So I'm just going to briefly give you an example of a few of these other genes. So short root 1, again, has an expression pattern similar to scarecrow, highly expressed at the base, turns off towards the tip once it matures. And it's enriched in the mesophyll, but during this laser capture, you got some of the internal vasculature in there, so I think it's actually showing internal vasculature expression. And when we looked at the mutant, again, we find a more severe scarecrow-like phenotype. We have loss of minor veins, here in the red triangles, distorted minor veins, here with the red arrows, adjoining veins with the blue arrows crashing together. And it also appears to disrupt internal vascular structure. So scarecrow only operated in the endodermis, but short root operates within the vascular core itself and the endodermis. So we see defects in both. The one thing we don't see is the extra bundle sheaths, because scarecrow is still operational there, so any signal still coming out can be stopped. So that's why it appears that these are more open as far as the vascular structure. So short root appears to have a conserved role as well. The indeterminate genes, this is indeterminate 1, which is previously identified as flowering time mutant, but it has high expression at the base, less as you go up towards the tip. And it's not shown in this figure here, but in C2s from the Colasanti group that first identified it, showed it has a high expression in the bundle sheath at the base of the leaf. And we find again that it has defects similar to the M2 allele, which are a little bit weaker, undifferentiated bundle sheath cells, colliding veins, and extra bundle sheath that adjoin veins. And the interesting thing with this mutant is that they were looking for flower time defects, but when they did a microarray analysis on the leaves, they found it had an altered expression of many of the C4 genes. So it appears that this is functioning in the scarecrow short root pathway as well to regulate identity of these cells. And one other one I'll show you here is ID-like 4899. It's pretty much undescribed at that point. And again, similar expression pattern, bundle sheath enriched. And it appears to affect the minor vein formation in this case. So we get a single cell layer bundle sheath, but it's really impacting minor veins. So just from looking at the conservation of the genetic pathway, we've identified many of the genes that are regulating Kranz anatomy. So when I started, we had no genetic model for Kranz anatomy. So now we're getting to the point where we can start the genetic structure. So we're proposing that short root, the signal that leads to endodermal identity in the root, is also the signal that comes out of the vascular core into the bundle sheath in the photosynthetic cells, activates scarecrow, and these two together lead to bundle sheath formation and identity. These probably interact with the ID likes, and we're still not sure, but there's a signal generated from these interactions that goes out to the mesophyll and identifies the C4 mesophyll type. And it's most likely going to be one of these scarecrow-like genes or ID likes. So I described in this talk so far the genes that have a bundle sheath specific expression, but within the scarecrow likes and the ID likes, this is scarecrow-like 3. They have a mesophyll specific expression pattern. So these genes are more likely to set up that initial interaction with the photosynthetic program where you form the malate in the mesophyll cells. So this is our model for the genetic regulation of Kranz anatomy that we've gotten from this work. Now there is one thing before the end of the talk I want to come back to is what's up with the grasses. So it appears that grasses and sedges are preconditioned for C4 somehow. And then in the dicots, it was the extension of the endodermal program into the leaf blade. But there's something different about monocots in general. They may not have true leaf blades. So according to the phyloid hypothesis, early monocots either have a reduction or great loss of the leaf blade, and we're left with a petiole-like structure. And the petiole became extrapolated into a new leaf-like structure, which gave rise to the parallel veination, and you don't get reticulate vein formation like you do in the C3 plants. So that would actually bring this program of the endodermis into the leaf blade itself in the monocots. And if we look at the early monocots, that the endodermis and the photosynthetic program is actually separated by parenchyma cells. But over time, these leaf-like structures became flattened, giving rise to the parallel veination. But in most of the monocots, you still have the endodermal program and the vascular core separated from the photosynthetic cells by those non-photosynthetic parenchyma cells. But it started, when you get close to the grasses, it collapses. And you get a loss of those parenchyma cells. And in grasses and sedges, there's no parenchyma cells. So the same event, morphological shift that led to the grasses and sedges, was the same event that preconditioned C4. And that's why you have the ancient preconditioning event. So from this model, we have the ancient preconditioning event. You could either select for or against C4. Again, it's a costly process if you need it or not. So in maize, it was selected for. Endodermis becomes photosynthetic bundle sheath. You get C4 metabolism and the architecture. You have selection against C4. It's more like we're seeing in the mutants that you get the endodermis becomes the mesotome or vascular bundle sheath, C3 metabolism. You still get that structure like there. It's an evolutionary, I'm thinking of more as an evolutionary remnant. And they're very similar to what I was seeing in my mutant when you lost the identity. And again, if you lost vein spacing, that would account for why you're getting increased vein spacing in this as well. So the thing to think about here is that it only took a few genes to disrupt the endodermal program and produce a rice-like structure. What's it going to take to get back? I don't think it's going to take 20 years, and I don't think it's going to take $1 billion. If we could just regulate this program, we might be able to get here pretty fast. And that's where we want to leave off, is that this doesn't look so far away anymore. So that, I'd like to acknowledge all the people who have helped me on this project. The Turgeon Lab, so Bob for allowing me just to dive into this project. And when I came to him one day and said, I think I figured out C4, he kind of just let me go, and I was glad for it. Ken Kui and Andre for all their help in the lab. Alyssa Anderson, a great undergraduate who helped a lot with the Scarecrow project. Collaborators, Tom Bertinel, especially Kevin Ahern in his lab for the field work, and Lin Wang for the RNA-seq, which I didn't have time to describe. All the members of the C3-C4 working group, Alice Barkin and the Uniform Mute Project providing all the mutant alleles, Rich Medville for help with the TEM, Adrian Roeder with the confocal, and funding from the NSF Plant Genome for the C4. And I am funded on a USDA NIFA postdoctoral fellowship to actually look at phloem loading in poplar, so I have to acknowledge them as well. And with that, I'll take any questions. Yeah.
Have you done, or has someone done complementation of scarecrow and shortroot and Arabidopsis with the maize homolog?
Yes, they have. So the first thing we did when we found this out was overexpress scarecrow and shortroot in Arabidopsis, see if it gave rise to C4-like structures, and it didn't. So the maize does complement it, but just complements it normally. There's nothing special. We don't think it's actually these genes that give rise to it, but the regulator of the whole process of repressing that program. So we're looking for the regulators that could just release the program itself. And that's probably why it would have been described by now of all the people working on shortroot and scarecrow that just overexpression in any plant would give rise to it. We think it's the whole program needs to come at once. So the regulation of that program is what we're looking at. Yeah?
So across maize germplasm, do you see natural variation for Kranz anatomy?
Not too much. Not too much. You can see a little bit in, sometimes you get a decreased veination or some extra bundle sheaths, but not a lot. So, yeah.
So Tom, if you look at patterns of expression of these genes in rice, like in the leaf Like,
Yes.
are they different than what you see in rice?
It appears to be turned off, like the scarecrow at least, is not expressed in rice leaf blade.
I don't.
It appears no. So it's on in the roots itself. It appears that the endodermal program is functional in the roots, and it's on in the guard cells, and that's about it. Yeah.
Completely naive question, which is, are there C3 and C4 plants that are closely enough related that you can cross them and look at segregating progeny?
Yes, Cleome and Flavaria, but the problem is with that is like there's not well-built mapping populations or markers for it. And Flavaria, a lot of times they become sterile. And with Cleome, they have built them, but I think they're trying to analyze them now. So, but those are the only two so far.
So those are those grasses?
No, they're dicots. Yeah. Yeah.
It looks like in your model that the short root is non-cell autonomous.
Yes.
It is signaling from the vein to the endodermis. And so what is encoded by short root? And, or maybe I missed this. So how does it work?
So short root is expressed just in the stele, but the protein moves from the stele to the cells next to it and initiates the endodermal process. So a lot of the identity is attributed to short root itself. So it's the protein that moves.
And the protein, can you tell what it's similar to? Or?
It's actually, as far as the lineage, it's in the GRAS transcription. It's called GRAS, transcriptional regulators, which encodes the scarecrow and short root. So they're their own clade in that respect. So.
Is it known or is it speculated why the monocots lost the leaf blade?
They think it had to do something with the aquatic origin of monocots, where it's either the upper leaf blade became greatly reduced when it went into the water and just became like a petiole-like structure to avoid being, drowned out, essentially. But I think it has something to do with the aquatic origin, a lot like Acarus, one of the original monocot-like plants. So they think the leaf blade may still be there, but at the very tip. And we were actually looking at the phyloid hypothesis for phloem loading because it appears all the monocots don't load raffinose and stachys or are able to polymer trap because those are minor vein specific programs, which may be lost in the monocots because it's specific to a leaf blade. So it appears to have something to do with aquatic origins. Yeah.
The sort of missing link, I mean, though it may not cost $1 billion in 20 years, I mean, it's still not simple, right?
It's.
And so, I mean, there's still more than one gene that you need to change to get there.
There may be one or two.
Okay.
We already have a hint of what genes are regulating it, and it's actually pretty simple. It is a simple change. It could be one or two genes that are just releasing it.
So there is no missing link. I mean, it's not for failure of having looked carefully that we can't find anything that's sort of halfway between a C3 anatomy and a Kranz anatomy.
Well, they do say that there's intermediates, but those have just been recent events that have led to, that have released the endodermal program into the leaf. It may not lead to perfect C4, it may still need more selection, so there is an intermediate type. But it appears still fully formed, almost 90% of the way there. But it appears that it's either one or two genes or a few genes that need changed, not many. Yeah.
So I know they've been looking for mutants in rice that would unleash this program. And if it were one or two, you would think that just through mutation, since it's a knockout, right?
Yeah.
That if rice were as similar to maize as you're suggesting, that it would already have been found. So I have two questions. One, I know that the group in Taiwan had found something they thought looked intermediate in terms of the number of veins and the distribution of cells between them.
Yeah.
So does that look promising? And alternatively, is the alternative hypothesis that maybe the reason why that occurs so frequently in, say, panicoids, but not in the Bambusoideae, that there's other genes that have interfering relationships with this development in some of the families of the grasses and that aren't necessarily there in others?
Yeah. So with the rice actually is more complicated because it may have already been preconditioned C4, but lost it. So the pathway may have been degraded at some point. and it's not as easy to reinitiate it like it is in the dicots. So by that model, it's easier to initiate it into dicot than it is in rice, because we're not sure if it had already been preconditioned and then undergone degradation, which is the harder part of this. And with, as far as like what they found in the rice and the activation tagging, we're looking for repressors of the program, so which release it.
Dominant repressors?
Yeah.
Okay.
So it appears that it's a dominant repressor, and most of the genes that we're looking at release repression somehow. So it's harder to activate. If you activate a repressor, you're putting it back on. So it's harder for that relationship to work. And with the other grass types, we're unsure like how the other genes would work, or whether again it lost the program at some point or suppressed it again. So with the bamboos or other monocots itself. So we're not sure exactly what's going on with those.
In other words, the point is really, going back to John Luke's question, that you may be able to diagnose it as a source in this endodermis, but it may not be so easy to trigger it
Yeah.
in the plant species that you want, either because it already occurred and is no longer even vestigial, or because there's some other differential regulation.
Yeah.
So I still think we are a long way from
We're
being able to move it, in a sense,
Yeah.
from one species to another.
It's going to be a little bit more difficult in the grasses. I think it's easier in the dicots, but as far as the 250 transgenes and the chromosomes, I don't think it's going to take that.
Okay.
And I don't think it's going to be much more to reawaken the pathway in rice than it is as in the dicots as well. So I think looking at the program, like the entire endodermal program and the root program is exactly similar to maize and rice in the roots. So that it's there. The whole thing is we just have to learn how to pull it back up. So
one other question, I'm sorry.
No, it's fine.
Just thinking about the bamboos. So we have annual bamboos. They produce biomass, far, far, far greater biomass than something that's trying to produce grain.
yeah,
So again, if biomass is your indicator, it may be great for biofuels.
Yeah.
It may not be great for grains.
exactly. So
Again, you look at the productivity of the rice plant, and as puny as it looks in your picture, it does a damn good job of producing.
exactly, yeah.
We just got biomass full of silicon.
Exactly. That's why I said at the beginning, we can produce the trait that then needs to be worked on to produce the grain.
Would need to be worked on.
And I think Mark Cooper put it best to me the one day I've talked to him, he said, the most productive and the least productive variants of maize are all C4. So he's really advocating breeding. It gives you potential for breeding. It's not the silver bullet. So you do have a lot of selection left after it occurs. Yeah, so yeah, I agree with that.
Geneva? There are no more questions? No more questions? Let's thank Tom again.
Thank you.